So, as we pick up here in chapter 19.2, we want to remind ourselves about some things about viruses. Viruses are obligate intracellular parasites. Obligate means that they have to live within another cell. So, obligate, you have to, intracellular, they have to be inside of another cell. And parasites, meaning that they are going to take over the host cell and use the host cell's machinery in order to replicate. Each virus is only able to replicate within a host cell, and this is specific to each virus type. Each virus will have a very specific host range. Some viruses are able to infect multiple animals or multiple organisms. Some viruses are only able to infect certain animals. If you think about something like HIV, right, it has a very particular host cell, a particular type of white blood cell that it can infect. What a virus does is once it has infected a host cell is that it'll use the host's enzymes, ribosomes, tRNAs, amino acids, ATP. It'll use all of your stuff that you have, and it'll take over the cell, and those viral components will start being spontaneously made and self-assemble into new viral particles. So here we can see a picture of sort of a basic generic viral replicative process. So up here we've got our virus, and you can see there's that virus, the DNA inside, looks like a double-stranded DNA virus, and then the protein capsid coat. When that virus gets into the host cell, that's this yellow part here in the middle, what happens is the capsid will open up, and the DNA will then be inside the host cell. And what this virus will do now is it will take over the host cell's machinery to start doing transcription, translation, and replication. Those viral particles will then spontaneously self-assemble, and as they leave, they can go out to infect new particles, new cells. Here's a nice image that we can see of a high school map. So those of you guys that are still in high school, sorry, it's really gross there. But just showing you how quickly viruses can spread. So this shows a single high school over a span of 18 months. And every dot is either a boy or a girl at the school. And then the lines represent places where viruses could be transmitted between them. Blue lines represent more recent transmission. Lighter gray lines are those that are in the further past. And as you look at this picture, you should start to get a little bit disgusted because it turns out there is not one single individual in this high school map that is broken apart from all of the others. And so what that means is if just this one person right here is infected with some virus, potentially the entire school is exposed to that same viral particle. And that's one of the reasons that you're seeing so many school closings right now as we're dealing with the coronavirus spread. As we think about how viruses go through this replicative process, we're going to specifically start by talking about bacteriophages. Remember, bacteriophages are these little alien-looking guys. These are viruses that infect bacteria. They're a good tool for scientists to use. We know a lot about viruses by studying these bacteriophages. So we're going to use this virus sort of as our model organism to sort of see some ways that viruses replicate. Phages, these viruses that infect bacteria, have two basic reproductive mechanisms. The first is called the lytic cycle. The second is called the lysogenic cycle. We'll go through both of these cycles. You should be comfortable with both. In the lytic cycle, what will happen is the virus is going to infect the host cell, and eventually what's going to happen is the host cell is going to die. The host cell is going to die by lysis. It's going to explode open. It's going to break open, and that will release the new progeny viruses. Phages that reproduce by this lytic cycle alone are called virulent phages. So on the next slide, we'll look at an image of this. We've got our virus attacking that bacterial cell. The first thing that that virus does is that it inputs its viral DNA into the bacterial cell. So that's that little blue piece of DNA that's now. What we can see happening is new viral particles are being assembled. Again, the virus is using the bacterial cell's enzymes to do this. That synthesis will continue. I do not worry about the steps of this, but we do see that those new viruses are composed. And then bad news for the bacteria, explosion or lysis. The bacterial cell explodes, lyses open, and now all of these progeny viruses can go out and infect additional cells. So again, this is called the lytic cycle. If you're a virus that reproduces primarily by this lytic cycle, you are called a virulent phage. The second basic type of life cycle that we see, not life cycle, reproductive cycle that we have, is called a lysogenic cycle. In this case, what happens first is that the viral genome is going to be copied without immediately killing the host. So at the first, it's going to look like it's a much better option for the host. The virus DNA molecule gets gets incorporated into the host cell's chromosome. This integrated viral DNA is called a prophage. And so what happens is the virus has attacked the cell, it's integrated its virus into the cell, but it hasn't taken over yet. It's just inserted its genetic information and it's just sitting there and waiting. Every time the host cell divides, it's going to copy that viral DNA and pass that copy on to daughter cells. And this will keep happening time after time after time, which right now probably looks pretty good for that host. However, when there is some environmental trigger, think something like there's not enough food, there's a poison, it's too hot, there's some sort of stressor, all of those cells that have that piece of viral DNA in them can then be switched or triggered to switch into the lytic cycle. Phages that reproduce sort of through this cycle by using this lysogenic cycle and then the lytic cycle, they are called temperate phages. And again, we'll look at these on an image on the next page. So let's start at the beginning. Here we've got a phage that's infecting a bacterial cell, and here you can see it infecting that bacterial cell. So we go to the middle, there you can see the viral DNA, the blue part, in with the bacterial DNA. Unlike the lytic cycle, in the lysogenic cycle, what happens is the viral DNA gets incorporated into the bacterial DNA. So you've got that brown bacterial DNA with a little piece of blue DNA in it as well. And this cell is not being taken over right away, right? It's still able to function as it normally would. Every time it divides, you can see that as it divides, that prophage, that viral DNA is being replicated. That's a good vocabulary word to know. Again, a prophage is what we call it, where we've got that bacterial DNA with the viral DNA also incorporated. This replication process will continue on and on and on until there's some sort of environmental trigger. When there's some sort of environmental trigger, again, just think some stress, that's usually the trigger. What happens is those prophages, they will drop, they will export, they will exit, they will no longer be integrated. And so that viral DNA is opening up. And now what happens is bad news. Now that cell is going to go through the lytic cycle. So it's going to take over the cell. We're going to take over the machinery to assemble new viral particles. And then those viral particles are going to explode out. So in this lysogenic cycle, again, it seems to start off fine, right? It's integrating, it's not impacting the cell. But then what's happened is that prophage has been maybe replicated to 2 cells or to 10 cells or maybe even 1,000 cells. So when there's a stress, it's not just one cell that's going into the lytic cycle. It might be all 1,000 or some high number of cells. Again, if you reproduce through this type of mechanism with a lysogenic and then a lytic cycle, you are called a temperate phage. This is a video that I encourage you to watch if you have any questions about CRISPR. It's a nice introduction to the next topic that we're going to discuss. So it turns out that bacteria for a long time have been getting infected by bacteriophages, and so they have some defenses against this. One thing that bacteria do is that when they recognize foreign DNA inside of themselves, they have special enzymes called restriction enzymes that will cut up any of that foreign DNA. Another thing that bacteria have done to evolve sort of a defense against viruses is they have something called the CRISPR-Cas9 system. The CRISPR system stands for clustered, regularly interspaced, short palindromic repeats. No one's ever going to ask you that long name, but that's what CRISPR stands for. What happens is bacteria have started remembering what different viral genetic elements look like. And so what happens if we look over here at this image on the side of the page, here's our bacterial cell, that's the yellow thing. In the bacterial cell, in their genetic information, they have what's called the CRISPR region of DNA. What they do is they remember DNA from previous infections by different viral phages. So you can see that they've remembered being attacked by a blue virus. They've remembered being attacked by this green virus. They've remembered being attacked by this yellow virus. And in this CRISPR region of their DNA, they have the spacer regions, those are the part in brown, interspersed with these sort of memories of previous viruses that have tried to attack them. What a virus also has are not viruses. What bacteria also have are these things called CRISPR-associated or Cas proteins. This is a nuclease. This is an enzyme that's able to attack and cut apart DNA. So here's how the CRISPR-Cas9 system may work if we look back at our picture. So we have a virus here at the top that's trying to infect this bacterial cell. It's injecting its blue DNA trying to attack the bacteria. When it injects its DNA, this activates the CRISPR region in the bacterial's DNA. And what happens is you can see that we make this RNA transcript down here that has the complementary RNA, that blue piece that would match the blue DNA coming from the virus, plus that little hairpin. From here, each of those RNA strands is going to bind to the Cas protein. That's that purple blob in the background forming a complex. This Cas protein RNA strand will then move around in the cell. So it's going to zoom around, and it's going to look for DNA that it's complementary to. Here we can see in this middle picture that it's found complementary DNA. What happens is when the Cas protein RNA complex finds the complementary DNA and binds, that Cas protein is a nuclease. It's able to cut DNA, and so it's going to cut that virus DNA. Now that that virus DNA has been cut, the virus DNA can no longer replicate, and it's going to be degraded. What scientists have done is they've taken this CRISPR-Cas9 system out of bacteria, and they've realized that this can actually be used as a tool to edit genes in various organisms. This is discussed in your book in a later chapter. It's on pages 424 through 425. So if you want to read more about this process, that's where you'll find it in your textbook. What happens when you use CRISPR-Cas9 as a gene editing tool is you're using this system that already existed in bacteria, but you're using it to alter an organism's DNA in some way. In order to do this, you need a couple components. So if I wanted to edit your DNA, if I wanted to maybe fix one of your DNA genes or give you a new DNA gene, what I would do is first I would need to have a guide RNA that I would engineer in the lab. So in the lab, maybe I'm trying to give you a better gene for blood pressure, right? I'm trying to fix your DNA so you don't have bad blood pressure. In the lab, I would have to make an RNA strand that's complementary to the gene that I'm trying to fix. In the lab, I would also need to acquire a bunch of the Cas9 protein. And then in the lab, I would mix those two things together to make this component. Once I had the guide RNA plus the Cas9 protein, I could then insert those into the organism. And at that point, the system is going to take over on its own. At this point, that molecule will move around and it will look for the DNA at any place that it is complementary. So here we've got our guide. And in the blue, you can see that it's found DNA that it's complementary to. And now that it's found that DNA it's complementary to, that nuclease, that Cas9 nuclease is going to cut the DNA. And so now I've cut your DNA. At this point, one of two things can happen. What I can do is if I'm just trying to turn off your DNA, by just cutting the DNA apart, your body is going to try to repair it with random nucleotides, and you are going to knock out the gene. Sorry there. So that's option A. Or if I'm trying to actually fix your DNA, what I can do is I can put in a template in addition to this process, and I can use this template to repair the gene so that I can get some sort of fix. This CRISPR process had not been used in humans up until November of 2018. Hu Jiankui, a scientist in China, used this process in a variety of human embryos. The two most famous are Lula and Nana, twin girls. What he was trying to do was he was interested in individuals who were being born to parents who might be HIV positive. And he wanted to come up with a way of protecting their children from being born HIV positive. He sort of was interested in the same process that we talked about earlier this semester, where we talked about how some people have that gene mutation in that protein on the surface of their molecules that makes them resistant to HIV infections. And so what he wanted to do with these baby girls that hadn't been born yet is he wanted to go in and change their DNA. He wanted to take their DNA from being the normal DNA that expresses those proteins on the white blood cells to being DNA that would not express those proteins, that would make them resistant to HIV. And he went ahead and did this without really any support from the scientific community. This was done in secret. Nobody knew he was doing it. And then after the girls were born, he announces this publicly in November 2018. Since that point, the entire scientific community has been very disappointed. It turns out there's a whole bunch of problems with this. Number one, we're not quite sure that the parents understand that their children's DNA is permanently now edited. That as these girls go on to have their children, their DNA will also be edited. Number two, the university where he was working, they did not approve this research. It was not under the protocol that he was built under, so he's been fired. They've distanced themselves from all of this type of research. Another problem with this process is that it wasn't actually that good. CRISPR is still a new technology, and so as we look at these two girls, instead of being completely protected, they are instead mosaics, where this process worked in many of their cells, but not all of their cells. So unfortunately, it didn't even do what we were hoping that it might do, or what he was hoping it might do. It didn't protect them. And then lastly, really important to point out, is that we already know how to protect children that are born to HIV positive parents. There's already a really robust drug protocol that doctors can follow. And so if parents are HIV positive, they put them on this drug regimen, and children are born all the time to HIV positive parents without developing HIV themselves. Again, this is a big thing in news because this guy really did something unethical, and scientists are still trying to play through the results of what's happened. What do I want you to know about this whole process? I need you to know how the CRISPR technology works. I need you to understand what the Cas9 molecule is. I need you to understand the importance of that guide protein, that guide RNA. I also need you to understand a little bit about what this particular scientist was trying to do in his studies. Again, two great links to watch if you want to learn some more about his particular involvement. As we think about viruses, one type of viruses that we want to talk about are called retroviruses. Retroviruses include an enzyme known as reverse transcriptase that is used to allow them to copy their RNA genome into DNA. HIV is a virus that you're probably very familiar with. It causes AIDS. That's the disease. In this virus, what it does is it goes sort of backwards. Up until this point, we have said the following: DNA makes RNA, RNA makes protein. And this is how most molecules work. This is how life works. This is sort of our flow of genetic information. What these retroviruses are doing is something completely bizarre. These retroviruses are starting off with RNA, and then they're using their RNA to go backwards to make DNA. And then from that DNA, they make another RNA molecule that's then used to make a protein molecule. So they're going in this weird direction where they're taking that RNA and going backwards to make the DNA first. We'll look at this in an image on the next slide, but you should be comfortable with sort of how the HIV particle works in this fashion. Here we've got our HIV particle. You can see the HIV particle includes the capsid, the RNA. Those green dots are the reverse transcriptase enzymes, and then it's surrounded by a membranous envelope. When the HIV particle infects the host cell, what happens is that reverse transcriptase will take the RNA, that red piece, and then start going backwards to build DNA. And so now you've got this viral chunk of DNA in the cell, and that viral chunk of DNA from the HIV particle gets permanently integrated into the human's DNA. This structure is called a provirus. This is a permanently integrated portion of the human's DNA. It's never popping out. It's going to be there forever. When the organism, when the human gets stressed in some sort of fashion, that provirus will get activated. DNA will make RNA, RNA will make proteins, and what happens is now we've got new HIV particles that can go out to infect the organism. In concept 19.3, we want to talk about the diseases that can be caused by viruses. Viruses definitely can affect humans. We're all very aware of that right now. They also, though, affect crops and livestock. Viruses, what they do is they often kill cells, and that causes the release of maybe hydrolytic enzymes from lysosomes. Remember we talked about lysosomes, there were those packages of acid. If those just explode everywhere, that can make organisms really sick. Viruses sometimes have envelope proteins that are toxic. Sometimes viruses also cause infected cells to produce toxins. Viruses, we can see two examples down here. This little guy down here has smallpox. This guy over here, he's not just chubby with freckles. That sort of chubby cheeks, red speckles everywhere is indicative of measles. Most of you guys have probably never seen an individual with smallpox or measles because these are both things that we can get vaccines for. I want to talk a little bit about what a vaccine is. So if we think of our picture of what a virus is, I'll try to draw one here. Here's the capsid, and then we've got some piece of genetic information on the inside. With a vaccine, what we do is we take that virus and we either erase the entire DNA element or we erase parts of the DNA elements from the virus so that it's harmless. It still looks like the virus on the outside, but it no longer has that genetic component on the inside. And what we do is we inject that harmless derivative into the human. When that harmless derivative gets into the human, that stimulates their immune system and allows them to learn what the virus looks like. Our immune system is quite smart. What our immune system does is there are special cells that their job is to remember things that they have been exposed to in the past. So what a vaccine does is it puts this weird blue little diamond virus outside bit into the organism, and then that stimulates your immune system to know, hey, this is a virus. Then, years down the road, if you are exposed to the actual live virus that now has the DNA in it that could make you sick, your body is going to be quickly able to attack this and you are going to be able to recover. Vaccines absolutely are super important to preventing certain viral illnesses. A good thing to remember about viral illnesses is that they cannot be treated by antibiotics. So if you get an earache, that's oftentimes caused by a virus. You can't get an antibiotic to help you that, like flu. You can't go, that's a virus illness. You can't get an antibiotic to make you feel better when you get the flu. Antiviral drugs, they help treat, but they don't cure viral infections. So something like a Tamiflu, what that's doing is it's trying to slow down the viral replicative process, but it's not getting rid of the virus. In general, viral vaccines take anywhere from about, on the fastest track that we know of currently, they take about 18 months to develop. And so as we think about coronavirus and developing a vaccine, that's probably at least a year and a couple of months out at its earliest possible onset. Some other things to mention about vaccines. Vaccines are really important for those of you guys in the health industry or that are thinking of getting into health industry. You will be required to be vaccinated and you will be required to talk about vaccine with your patients and those that you come in contact with. Here are some things that I often hear said about vaccines. Number one, I don't get the flu vaccine because whenever I get the flu vaccine, I immediately get sick with the worst flu ever. A couple of things to just make sure we're really clear about this. Number one, vaccines such as the flu vaccine do not cause the flu. what happens is when we get our flu vaccines or any vaccine, it takes your body about 2 to 3 weeks in order to build up its immunity. So if I get vaccinated on Friday, I'm going to need to wait about 2 or 3 weeks until I'm actually learned how to fight off that vaccine, fight off that virus. A lot of us, though, are impatient. So we get vaccinated on Friday for the flu, and then our friend calls up and says, "Oh, don't come on over. I've got the flu. Don't visit me." And you're like, "No problem. I just got vaccinated." And you aren't actually immune yet. So we have to be really smart. After you get the vaccine, you need to give yourself 2 to 3 weeks of really good time in order for your immune system to learn and develop that immunity. Second problem is vaccines and immunity are really related to stress. If your immune system is under a lot of stress, so you're not eating well, you're not sleeping well, you're not taking care of yourself, maybe you're sick for another reason, your body won't remember as well. And so you want to make sure that you get vaccinated when you're in top health, not when you're already feeling slightly sick. Vaccines are really important. They definitely work. You should be vaccinated every year. You should get your flu vaccine every year, and you should be up to date on all your vaccines. Emerging viruses are something like the coronavirus that suddenly become apparent, things like Zika and Ebola, or Corona is better to talk about it. Corona, if we think about its actual name, the virus name is SARS-CoV-2. The disease it causes is COVID-19. How did this virus appear? Despite whatever crazy conspiracy theories that you may have heard, it's just mutation. This virus is a virus that existed in another organism. As it mutated, it jumped hosts. So instead of being able to infect just something like a bat, it was then able to jump and infect humans. And as we've seen in the last month, viral diseases in small isolated populations can quickly spread around the world because we're so interconnected. Most of the time when a new virus emerges, it is exactly this process that we've seen with the coronavirus, where we've had an animal virus that's jumped into humans, and because it hasn't been in humans before, our immune systems aren't good at fighting it off. In 1918, 1919, there was a bird flu virus that did this. It caused a pretty bad die-off in that year. About 10 to 20% of those who were infected died. Over a third of the world's population was infected. Anywhere from 3 to 6% of the global population died that year. It may have killed as many as 25 million people in its first 25 weeks. And you can see very scarily what happened that year is it was killing people in the prime of their life. And this is one of the reasons people are being so cautious about coronavirus and trying to really minimize the exposure, especially because you can see these numbers that we've had in past emerging diseases are very big numbers, right? That is not a small amount of individuals. In addition to affecting humans, viruses can also make plants sick. There are over 2,000 types of viral diseases in plants. Most plant viruses have RNA genomes. Nobody's going to eat these tomatoes. Those are disgusting, right? But people often pay extra for gourds or tulips that looks like this. And both of these conditions are created by viruses. So some farmers will purposely infect their crops with viruses because they're trying to get some of these added shapes. How do plants get a virus, or how do you make a plant sick? They can't cough on each other. So there's two ways that we spread plant viruses. You can use horizontal transmission or vertical transmission. Horizontal transmission is when plant viruses spread horizontally. Imagine within the same field. So what's happening is maybe we've got a big hailstorm and that's knocked over a bunch of the plants, and viruses in one can get to the next, can get to the next, can get to the next. We also have vertical transmission. Vertical transmission goes from parent to child. So if mama plant has the virus, when she makes her babies, those seeds will also have the virus.